So with no words, so you have one more example of what a short paper presentation talk looks like. And also you do want to tie in a world from dust. I do have points where I try to do that. So there we go. Okay, so that works, kind of. All right, so the thing that we want to talk about, so I have 3 parts. We're sort of going to have more biology in part 1, biology and physical chemistry in part 2. Part 3 is going to be pretty much just physical chemistry, but direct connection to the chapters we've talked about. So let's start talking about biology. How can a chemist look at organs and look at those? So the first thing is, of course, mitochondria. I don't know how it became a cliche to say they're the powerhouse of the cell. It must be in some high school biology book. But yes, mitochondria are pretty clearly distinct. They even have their own genome. And so the theory about how did we get these little bubbles that are the size of bacteria and are even genetically distinct like bacteria are, they even have circular genomes like bacteria. So the idea is that a long time ago they were bacteria. And chloroplasts look sort of the same, but it seems like mitochondria are more fundamental. So the idea is that the endosymbiosis is how things originally came about. Oh, the other piece of the puzzle, there are certain cells that eat other bacteria. So once you have a cell that eats another bacteria, and perhaps it couldn't digest it, it like stuck around like too much gluten or something like that. And then it actually set up a way to trade materials back and forth. And so instead of eating it, it's sort of like, they hung around and they were sort of, they formed a partnership. And the idea is that you can do that with bacteria. The evidence for this is pretty good, but it's also very old. So there's a lot of this that has been lost. But I think the basic chemical idea is that you can form partnerships like this. And if that's the case, then there should be some chemical reflection of when the partnership occurred is how it's mediated from place to place. And just remember that iron and manganese were used for certain things a long time ago, but then it got harder to use iron and manganese in the +2 forms. Manganese became more useful in the +4 form. Iron in the +3 form is not useful at all because it's solid. So basically what you have is you have this idea that as these different things came online, the elements they could use at the time were sort of locked in to mediate the partnership. And that's one of the reasons why calcium is really important for eukaryotes, is important for navigating this, because it's actually at the stage where iron and manganese 2 are less useful, and you want something that is exterior to the bacterium. The bacterium needs to communicate with the rest of the eukaryotic cell, right? The mitochondria. And so the most logical thing is something that the bacteria excludes, which is calcium. And yet also something that the eukaryotic cell can grab onto, can include. And calcium actually fits the bill for that. So there's actually a chemical reason for the reason why calcium spikes are what mitochondria use and do. Hey. The other thing that you'll notice is notice how zinc comes along, especially when you get to multicellular organisms. The other thing that I would like to say, a lot of times I talk about how copper comes in late. But this, even from R.J.P. Williams, this is an R.J.P. Williams figure from long ago. He even has copper showing up with the very earliest eukaryotes. And so, yeah, copper is later, but there's a lot of copper doing things even in the Middle Ages of life. So it's probably better to focus on zinc than on copper. And if you remember from the previous data talk, the copper data does have some ambiguity around it. And I think that now that I look at R.J.P. Williams, I'm seeing that he was saying something like that. Okay, but all that means is also copper is used for signaling on this end of things. So copper and zinc on the right side of history. The left side of history has iron and manganese too. So these could be considered the first organs. You know, before you have multicellular structures, you do sort of have inside a cell, you have subcellular structures. And just remember for those who've been through the biochemistry too, mitochondria, they make ATP. The partnership is by you give the mitochondria stuff to oxidize. They oxidize it and they give you ATP. And the chloroplasts actually do the reverse chemistry in a sense, but they do it by capturing light energy. So the mitochondria release energy, chloroplasts absorb energy, but each of these has their own even circular DNA. To me, it's the circular genome that really seals the deal. You can even look at their own membrane. It has bacterial-type lipids. To a biochemist, those look like bacteria that are living inside your cell, if you look at their chemical compositions. So this looks like it was the first kind of specialization inside a cell. But then you can get cells sticking together, and then you can get one whole cell doing a job that another cell doesn't have to do. So remember, in all of this, the cytoplasm is still reducing. Even in your own cells, the cytoplasm itself, it tends to be the same reducing environment that we've always had ever since at least eukaryotes were first invented. It reflects the old Earth. And I think this is another fascinating piece of evidence. A lot of the organelles are oxidizing. There's even an organelle called peroxisomes. So it seems like the organelles are the stuff that the cell would otherwise eject. But instead of ejecting it, it's putting it into a little bubble inside itself, which you see is kind of the same thing as ejecting it. If it's all about the cytoplasm, you're just getting rid of it from the cytoplasm. So you see how this can develop complexity both in what you put outside the cell and what you put into a closet inside the cell. And there's the same pattern. The most reducing environment is the cytoplasm itself, and the other environments are more oxidizing. So this is one of the ways that a chemist thinks about biology. Again, these are from R.J.P. Williams, and I do want to give him credit because all the ideas are his. I guess if there's something proven wrong in the book eventually, I can say it's not really my idea, it's R.J.P. Williams. But for right now, I'll let him take all the credit too. So they need to communicate, and if you look at how things communicate, it looks like you have a progression that fits with the chemical progression of the Earth in the kind of communication they're doing. Eukaryotes use calcium, which is actually not the oldest thing, but it's also not the newest thing either. But multicell eukaryotes will use zinc a lot, and they'll also use some other things. Has anyone taken a class like where you talk about neurons from a biology perspective? I don't know how much they get into it in cell bio. There are neurology courses. Has anyone taken an actual neurology course here? I haven't. But I know that there's something, and I know that, of course, Ridgway works on neurons, and he actually has confirmed these things. He even did some experiments with zinc that basically went right along with what the chemistry says that these should be. So even though these are old things, this is what Williams figured out from the way stuff works. And if you figure out stuff from the way it works, you have a pretty good chance of getting it right. As long as you have a complete view of it. So just look down here, when you get to multicell eukaryotes, then you're using the single charged things to signal. This is not because these are not redox sensitive, and it's not like you have more of these all of a sudden. But you can use these to make faster kind of signals so that you're only ready to signal in this speed when you have complex multicellular organisms, basically when you can have brains. Brains are specialized organs that move very fast, and they use fast elements to work with that. So notice how calcium is there, and then you have the single charged ones, which are super fast, which only show up when you have brains. In the middle, you have sort of long developmental processes. As you're growing, everyone talks about hormones and things like that. Hormones are small molecules we can make with organic chemistry fairly easily. And we can use them to send the same signals. But notice that these are slow signals. It takes time to piece together carbon. It takes time for it to float through the body to the other thing. And so it works well for organ-to-organ communication. It does not really work for something that needs to be fast like a brain. But realize that all of these are communication. They're just communication with different kinetic rate constants. So there's a couple of cool organelles. There's actually a lot of organelles, and it seems like they're always finding new ones. This is an interesting paper that just came out, accepted 31 March 2023. But what do you notice about when it was received? What do you conclude if you have received in May 2020 and accepted in March 2023, 3 years later? What does that say about this paper? Any ideas? This is slow. I mean, scientists always complain about how slow it is. Usually that means a year or two. Does not usually mean 3 years. What does this make you think about these findings? And it's also, by the way, in either Science or Nature, I forget which one, but it's a very prominent journal. Why do you think it took so long? Why would it take so long for a paper like this to come out? It seems like the reviewers are skeptical, and the reviewers probably asked for lots of experiments to be sure. So the long time of acceptance, along with the prominent publication, actually makes me more certain about this, because it didn't just fly through peer review. They really tried to ask about it. And basically what it is we have a phosphate-based organelle. And the thing about this is they don't actually say this in the paper, but to me it makes perfect sense chemically, which is kind of the cool thing. So here's the idea, that when you have adequate phosphate, you take your extra phosphate and you put it into this organelle. So when you're starved for phosphate, then you can degrade the organelle and get the phosphate back out. It's not really very complex, right? It's just basically a storage place for phosphate. And it's built around the idea that you have a phosphate transporter, shown as the blue squiggle here. And but it's definitely, it deserves the name organelle. But chemically, it's pretty simple. I just want you to see that organelles can have pretty simple chemistries. This is just a room for phosphate. And when you need the phosphate, you open up the door and you let the phosphate out. The other thing is, remember, phosphate is related to calcium. You have to be very careful with your phosphate and calcium levels. If you have too much of either one, they will make little calcium phosphate rocks inside yourself. So it's good to not have too much phosphate around. And it seems like this organelle could have evolved for a case like that, but then the organisms would survive better because they'd be able to survive phosphate limitation, phosphate starvation. So here's another one, talking about calcium. Another thing that the cell often wants to eject, because after a certain level, after you get above the solubility constant, you start to form solid. And so you need to stay below that level, and so you need to eject it. And that's what calcium is. Calcium is the center of it. And as you know, calcium is in coral reefs and things like that. That's a consequence of cells ejecting calcium, and then it solidifies outside them. They're happy for it to solidify outside them. Well, this is an interesting thing about how did coral reefs evolve? Calcifying microalgae are like the progenitors of coral reefs, but there's cousins of them that are non-calcifying. And so what's the difference between them? Well, what they found is they both have very similar calcium storage organelles. It's just in the calcifying ones, they end up having apparatus to take that calcium and to put it outside in a coral reef kind of organization, calcium carbonate. And so this is a pretty cool thing. They're called acidocalcisomes, are these kind of organelles, and they're calcium containing. They've basically been seen even in bacteria. And by the way, bacteria do have organelles. They just have simpler ones. Okay. But they've been proposed to represent an intracellular calcium pool. But remember, they're inside a membrane, or they're inside a membrane inside a membrane, right? They are not really in the cytoplasm itself. So the cell controls what's in the cytoplasm by putting it into the organelle. And so what's interesting is that one of the questions you have to ask is, when somebody says, I found a new organelle, why didn't you see it before? I mean, organelles are the kind of things that you should see. But they say that they were they give you a reason that the calcium, these calcium organelles were overlooked because they're hard to see, and most people prepare them and they blow them apart when you're looking at a cell. But they did this, and I hope this works. Okay, good. This is a 3D construction, and this is the part of biology I wish I could do, but as a chemist, I just have to sit back. So they take all these slices, these microscopic slices of the cell, and they're imaging where things are. Eventually, you'll see the colors. The blue colors are the extracellular coccoliths. They're the calcium that's building into the coral. And inside, you see the red blobs. And you see all these other biological parts of the cell. But the important thing is calcium is outside in blue rings, and it's inside in red blobs. And they take away everything else, and you can see those red blobs, they're definitely intracellular. It's definitely going to take some kind of effort for you to get the calcium from the red and export it into the blue, and you can have some enzymes that sort of make the shape, the right kind of shape for the cell. But the basic idea is that you have these red blobs to start with, and the red blobs are a pretty easy thing to imagine evolving. And so I want to say that the wonderful thing that is a coral reef actually can evolve from the fact that if you have something you need to get out of the cytoplasm, you can put it into an organelle, and then who knows what can happen to that organelle's stored calcium. Someday you can figure out how to turn it into the blue coral reef structures. Starts out with storage, and that's the basic evolutionary concept we want to get here. So once you start putting things outside the cell, you can start building a coral reef, or it can be, if you're in a multicellular organism, it can send the signals to other organs in the body, and there's lots of organs in the body. In biochemistry, we actually don't talk about organs very much. But in biology, you'll get more of this. But I do want you to be able to put that biology into the framework of chemistry. And so if you have all of these things, you actually do have, and basically the less central the organ is to the organism itself. Like the heart is something very closely related to what it's a very old organ. The brain is a relatively newer organ. And more specialized organs, like things that only mammals have, and things that only the mammals and birds and things like that. Reproduction is very different, so reproductive organs are relatively new. The newer it is, the more likely it is to depend on a new element for signaling, which includes things like zinc and copper used for signaling. I want to be clear that this is copper used for signaling as opposed to copper used for catalysis. And even, by the way, the thyroid. Have you ever heard about getting a goiter? Like your thyroid swells up when you have a goiter. And the element that prevents goiters is iodine. And so sometimes people wouldn't get enough iodine, especially like a century ago. So to prevent goiters, they started putting iodine in a very common material that we all use. So what is the iodized chemical that you put on your food? Anybody know? Yeah, iodized salt, right? And that's literally, but iodine is only used for the thyroid, which has advanced signaling functions. There isn't really much iodine signaling done by any other system. And iodine is also a relatively new kind of element. And so, we don't have as much of a problem with goiters now as we used to, because we don't have thyroids. We get enough iodine for our thyroids to signal properly. But that's why it's just the thyroid, and that's why it's just this complicated signaling system. It's definitely a multicellular signaling system. It uses a new element. So you look up and down, you can organize it, and if you look at how fast it needs to work and what it needs to do, does it need to chemically break things down or not, you can actually come up with a conclusion, a rough conclusion, for why certain elements are in certain organs. And I just want to, we can't go into detail on that in this class, but I do want you to have this chemistry filter that you can talk about. And by the way, like in a biology class, a lot of times just a simple chemistry filter will allow you to ask questions no one else is asking. So I want you to think interdisciplinarily, when you're going into other classes. You can also organize this by element. Say we have these elements. Which organs need those elements? And if you look up and down, what's the organ that uses the weirdest number of metals for catalysis? And I just basically see something that pops out to me. I wonder if you see it too. Well, basically look up and down the liver. And I don't know, I actually like liver, but maybe there's a metal deficiency that I address by eating it. But most people don't like liver. Most kids don't like it, probably because it's so rich in metals. It's also, it's got lots of things in it. But the liver has a ton of metals. That's why it's so nutritious for you. Because it does so much catalysis. It's kind of like the chemistry, it's the chemist of the body, in a sense. Because it breaks down stuff and it detoxifies stuff. And it uses a lot of these weird catalytic metals for that. And so there's fascinating things about that. On the other hand, things like selenium get in the kidney, and I don't think the kidney actually uses cadmium, but it's probably filtering stuff out. But if you look at zinc is in a lot of these. And so one of the things to think about is, why do organs, and there's one I want to do before I move on. Well, these are pretty much the catalytic metals. They don't even include the single charge metals. They don't include sodium and potassium here because they're not really in these other things. Notice that the brain only has a little bit of copper, but the brain has these specialized chemicals that are sodium, potassium, and chloride. And so what do we say about these chemically? Well, we can look at them as far as big and little k's. And so this relates exactly to chapters 6 and 7 of Atkins. When we talk about binding just to negatively charged things, these are mostly positively charged. So when these positively charged things bind negatively charged things, carbonyl groups, when they bind any form of oxygen like an OH or things like that, sulfur groups, in general, they have very fast K-ons. Look how big that number is, 10 to the 9th. And also very fast K-offs. And so big K is a ratio of the two little k's. And you can actually follow that ratio because R.J.P. Williams did his math right. And their binding value is actually pretty low, but they bind quickly and they let go quickly. On the other hand, calcium binds quickly, but it doesn't let go. It's got 2 charges instead of 1. It ends up with a much higher binding constant. And so it's not good for sending fast messages, but it's good for a fast-acting trigger that then stays on, stays bound, and doesn't let go immediately. Magnesium is a little bit slower, so it's actually good for even slower catalytic things like phosphate transfer. And then you have zinc. Zinc is very fast on, but it's very slow off. So you don't want to use that as a mechanical trigger because zinc would mean your muscles would never unclench. They'd clench really fast, but they wouldn't unclench. And so the idea is zinc is good for slower kind of things that are not on the scale of muscle movement, but on the scale of organs talking to each other. And so it's good for communicating from organ to organ because of this. And copper, it's really fast on, but it's almost unmeasurable how off. It never lets go. So it's not good for communication at all. It's like the signal that you can turn it on, but you can never turn it off. You have to turn off the signal at some point for it to be a good signal. And so I just want you to look at these and to realize that these are fast on, fast off. These are slower on, slower off. And then you have the slow on, slow off is magnesium, and copper is never off, basically. You can sort of say, how fast are the rate constants that each of these do, and the rate constant for binding is a good first approximation of what can you use it for in the body. So if you look at these rate constants, this is actually, the bigger it is, the higher up it is, the faster on it is, the farther down it is, the faster off it is, and just notice that sodium, potassium, and chloride are the fast on, fast offs. These other are not really good for fast signaling in the brain. Now you can imagine that maybe if you had a different kind of brain signal where the medium off of calcium would be okay, that would be used. But you want everyone to use copper for brain signaling. You'd want to use it for catalysis, but not signaling. So if you look up and down on these things, I just want you to understand them in terms of rate constants. And one of these binding constants is how fast does it bind water. We said how fast does it bind things that are negatively charged. Water has a partial negative charge on it. And so this goes along well with specifically how fast do they bind to water. And you can see that the water, we have the same kind of grouping, very fast binding to water, but magnesium is slower. And things that we don't use at all, like aluminum and beryllium, are so slow that they basically stick to water so tightly that using them in the biological context has the problem of getting the water off of them. So this is another reason why we don't use aluminum. So slow organs use slow elements, fast organs use fast elements. And you also do have other things. You have the neurotransmitters, of course, but as chemists, we're focusing on the elements today. And the thyroid, very slow, and here's the thyroid. It actually has a lot of, it's the only place where you really see iodine. And that's because the thyroid developed really late, and kind of all the other elements were taken. And also it works slowly, so it could use a slow element. And probably, I would assume that it developed in an ocean animal, because you have a lot of iodine in the ocean. In fact, that's where they get it for the salt. But that's why I want you to think in general. That's thinking about biology in terms of chemistry.